Christina, thanks for the invitation and for introducing me. My Spanish is so lousy today. It is literally inexistent, so I need to talk in English with you, and I hope you bear with me. Thank you. So we're going to talk about, or I'm going to talk about the deep brain stimulation in the depression network, and it's the network X aspect that I think is very, very important if you're treating diseases like this. Just some key facts. Depression is a very common and prevalent disease. More than 300 million patients are suffering from a form of depression at any time, and 1 million patients a year, 800,000 in those new numbers here, commit suicide per year because it's such a devastating disease. There are very effective treatments for depression. So most of the patients will never be treatment resistant and will never need anything like deep brain stimulation or anything else. And the effective treatments that we have are drugs, psychotherapy, electroconvulsive therapy, if you're starting to be treatment resistant, and then transcranial magnetic stimulation that is typically done in the repetitive or the deep version now. And if you are not reacting to any of those treatments, you might be one of those who might need something like a stereotactic treatment, and we're going to talk about that now. And the experts from psychiatry think that more than 20% of the patients are treatment resistant, but it's by far not those 20% that will need treatment with deep brain stimulation or anything. So Andres just came in the room, so I have the slide up for you. That's the Andres, of course, Andres Lozano and Helen Mayberg did the pivotal work on that, on deep brain stimulation for treatment-resistant depression. That's their first series where they basically showed in functional imaging that if you have a highly overactive CG25 and implant bilaterally in CG25, then the CG25 activity comes down, the metabolism comes down, and that coincides, and we knew that from studies that Helen Mayberg did before, that coincides with a very good treatment. At the same time, the metabolism on the right orbitofrontal region even goes further down, but that's just a detail. Now, it was even though Andres was the first one to do deep brain stimulation in larger trials. Then that's not the first. That's not the first series of operations that have been performed. I divided them by stereotactic lesion surgery here on the left, and then deep brain stimulation. And stereotactic lesion surgery actually started with the report of Spiegel and Weisses in 1947, where they already reported on 12 patients that they performed dorsomedial thalamotomies on as the aftermath of the psychosurgery that went on in the 30s, 40s, and 50s. And basically they did degeneration experiments and found that there was an emotion system in the depth of the human brain, and this emotion system basically you could address in the dorsomedial thalamus that is here. Now, they had some problems with the dorsomedial thalamotomies because patients went to be confused, and so that was left. And then other people like Leitinen and Lexell, they started Tallarach and Lexell, they started to do lesions in the anterior limb of the internal capsule. Later, cingulotomy was performed, and then subcaudate tractotomy that would initially done with yttrium seeds. So on the other hand, we have DBS. And of course, there's CG25. You can see how I deliberately moved that into the direction of where the subcaudate tractotomy is. So that has probably some influence on the same fibers that are going down there. Nucleus accumbens was done by Storm. Later, that was VCVS by Dougherty and co-workers. Bad nucleus of stereoterminalis and phyothalamic peduncle are used sometimes. Our medial forebrain bundle that we're going to talk on, and there's some literature on the lateral habenula, which has never really made it to a published clinical trial, although there's some hints to that the lateral habenula appears to be an important target. Where are we in the development of a therapy? So that's still a therapy under investigation. And in the development of a therapy, we have 2 trials that were both stopped, industry-sponsored trials, and I'm just going to talk about the CG25 trial by St. Jude Medical, which is now Abbott. And they did subcallosal stimulation, and 90 patients were included there, 60 active, 30 sham, and you can see that basically if you look at the MADRS responses, and I've tried to mark that a little bit, so that's where everybody is now stimulated, and this is the response line. So that basically didn't work, at least in the first 90 that were included, and then there was a futility analysis, and if you look at later analysis, you see that they're moving into the direction of a response, but certainly there was not a response in that, and so the trial was stopped or put to a permanent halt. Now the consequences are, they say, yeah, it's feasible, you can do that, it's safe, we knew that. And they're talking about maybe they are more chronically depressed than in previous studies, but some interesting thing comes up from the work that these groups around Helen Mayberg have done, actually. So they're now targeting with diffusion tensor imaging, which we think is very wise to do that. And what basically then, I'm moving back, sir. What they basically then found out in an independent analysis basically is that you need to use something like a tracked target, and you need to try and find the communication of those 4 tracks that you're seeing here, which is basically the cingulum bundle, which we think is the most important candidate, the forceps minor, and then you have here the uncinate fascicle. And if they do that, they did another independent analysis of that based on that work, and now 9 out of 11 patients, again a small and open label group, is now responding. That's 82% in the first year, and that's pretty reassuring. So obviously that is something you need to personalize that. You need to get a better understanding of what the depression circuits are, and that's exactly what we're also doing. So this is work that we're doing in our laboratory. That's not electrical stimulation. That's an optogenetic stimulation of a long Evans Cray rat. So we have a when it says on, somehow it's not working. I try to start it again. So at the moment when it says on, then the light is basically brought into the ventral tegmental area and it starts. And then when it is on, the rat starts to show a very, very typical seeking behavior. That's appetitive motivation. So that rat basically falls into a reaction that it would have. It would be seeking for food foraging. If it would be seeking for a mating partner and all that. The interesting thing is just here, the thing is empty, so nothing's there. So we are activating that system artificially, and we think that this is something that is very, very reminiscent of what's happening in those patients who have a deficient reward system. Now, the reward system appears to be very, very important for all of us. So it is a decision maker. It's a very, very important thing. I just had a discussion with Juan Garcia about the same thing. Dopamine appears to be much more important than we thought, although I do not think that the typical mechanism in the antidepressant response that we see is dopamine. But it's very interesting to look at the system itself. And that basically, that's the cartoon that was accompanied in the Nature paper by Howe et al. And the interesting thing is, if you have the idea of wanting something, then the free synaptic dopamine in the nucleus accumbens goes up. We know that if you do stimulation to the nucleus accumbens or the anterior limb of the internal capsule, you get an increased dopamine level. And that has actually been shown by the group from Damian Denise in the Netherlands with Rick Shorman as the neurosurgeon, and the appetitive motivation goes up, and neuroscientists will tell you the reward itself is not dopaminergic. The anticipation of reward, that's dopaminergic, but the reward itself is cannabinoid and opioid, right? And that's the least, although that's called the orgiastic part of that reward system, you need to have that to trigger that whole mechanism. But interestingly enough, it's the least important part, and that reward system probably should be renamed, and that is something that Jaak Panksepp did. Jaak Panksepp in effective neuroscience said it's the seeking system, reminiscent of that this reward system actually has more to do with the anticipation than with the experience of reward. Now, brains have developed, and probably we had like developmental steps. And the interesting part is that there's some animals here that, so brains have developed in multiple parts. So not all brains are the same, but it is fair to say that if you have a modern human sapiens brain, then that has had some pre-steps to it, and that you could look at pre-steps. That's why we do animal experiments. And I'm not saying that I have seen a depressed rat, but I've seen behavior that is reminiscent of something that could look like a predecessor of depression. And I think that's very, very important to look at that. Why am I saying that? So what we basically have been doing since 10 years almost now is we looked at patients that actually had SDN stimulation for Parkinson's disease, and we had one patient in which we had the luck to have a very, very good tractography image, and at the same time this patient developed out of euthymia without any form of depression or anything before that. We pushed him into hypomania. A lot of people who deal with SDN-DBS have seen that. That sometimes happens, and we now know that needs to be reprogrammed. You can do bipolar stimulation, steer the current into a certain direction, and that is a thing that most of the times can really be handled. And the idea at that moment in time, whilst we were describing the medial forebrain bundle, or what we now call the ventral tegmental area projection pathways, this structure here basically feeds into the medial forebrain bundle system, and the idea was then, how about we would start with a patient that had a depression, and maybe along the lines of depression is related to anhedonia, hopelessness, and all that, maybe a deficiency of the reward system, try to trigger the reward system. And actually that is what sparked off the idea of medial forebrain bundle stimulation for depression. Now, the medial forebrain bundle is a complicated structure. We knew a long time that there was direct connections between the ventral tegmental area, which is not anymore a part of the hypothalamus, which is sitting behind the hypothalamus. It's part of the midbrain, and that had a lot of connections to the orbitofrontal cortex. And interestingly enough, there appear to be fibers that go to the dorsolateral prefrontal cortex, to cortical structures that are reward related. And it appears to be interesting that if you are looking at, and I'm just letting that flash up, at the studies that had been performed at that time from Andres Lozano, Helen Malberg, Don Malone did a study on anterior limb of internal capsule stimulation. We had nucleus accumbens by Schlepfer and Storm. And you see that you have small numbers of patients. The largest one is 20 here, 20, 15, and 13, and always like a 50% response. And our idea was, why not stimulate where that whole fiber system comes from, very close to the ventral tegmental area, a little bit close to where we have induced hypomania in the Parkinson's patient, and that's actually the original idea of medial forebrain bundle stimulation. This target, for the ones who are ever interested in looking up where that is, the target was published and hidden in this paper. So that's the cross-species effective function of the medial forebrain bundle, where we also did simulations of how likely it is to touch the medial forebrain. I'm going to come back on that in a second. So medial forebrain bundle comes from the ventral tegmental midbrain. It has a mesolimbic and a mesocortical part. They unite, and then they basically move up to the frontal lobe. And then if you then do segmentations and look at where this are actually going, you see if you talk about part of the frontal lobe, you see a lot of dorsolateral prefrontal cortex. You see a lot of orbitofrontal cortex. And if you want to have that in Brodmann regions, in Brodmann area 8, 9, 10, 11, 11M, and some 47 on both sides, where basically that goes to. And these regions are typically also reward related. Here you can see it once more how that basically hits the frontal pole, actually, which is Brodmann area 10. Now, The problem with discussing with psychiatrists is psychiatrists work with other stimulation forms. They work with transcranial magnetic stimulation. They look at their fMRIs and stuff. So if you look at this very nice review paper by Lee and Friston, what you actually see is if you look at the typically the usual suspects that you have the effect network. You have the reward network, default mode network, cognitive control network, and they're all located on the cortical level. But however, we are dealing with stuff that we're doing in the depths of the brain. So how does that all come together? So our idea at that moment in time was, why not bring the two together and look at actually what's going on in the depth of the brain? And that's unreviewed data that I'm sharing with you now. So that looks rather chaotic here if you look at the prefrontal cortex, but you can see certain fiber distribution. They intermingle, but you can see some distribution. So there's a lot of green in the front here, and some orange here, and some blue up there, and some red in between. I'm going to show you on the next slide actually what we're dealing with, and that's now rather complicated. So what we did is we looked at 200 human connectome subjects, and did tracings from subthalamic nucleus, red nucleus, substantia nigra, ventral tegmental area, and then basically looked at where they are all projecting, and then try from the projection pathways and from what we could find in the literature from the subcortical parts, how could we address them and basically find a systematics that then goes into the three of the four networks that we're talking to, and because the default mode network is a connection between frontal and posterior, that's not something that we directly had a structure in the depths for, which probably is amongst others the cingulum bundle, but we did not address that because we were looking at projection pathways. And what you can see here already on this slide is that if you're looking at the reward pathways, which are the green ones here, and the effect pathways, that most of the stimulation points and some of the points where you do TMS and stereotactic lesion surgery are actually addressing those two pathways, not addressing anything else. And you would say, yeah, well, that's not a real surprise. But on the other hand, that has never really been shown in a way like that. So that's the fiber renditions of that. And I'd like to draw your attention up here. When you look at the anterior thalamic radiation coming from the mediodorsal nucleus of the thalamus, which is clearly part of the effect system and the effect network, and you look at the SLMFP or the VTAPP, how you want to call that, so the reward network, you see that they're basically addressing the same parts of the cortex, so they're converging onto each other. And that is something which I think is very important. You can find that in the others as well. Ventrolateral thalamus also converges somewhat more posteriorly. You can see the same thing for the STN. You can see that same thing for the nigra and for the red nucleus, while they are all addressing huge parts of the prefrontal cortex. Now, what we think is we need to look at the networks in a bottom-up fashion, how they developed, because looking from top down, how they are hierarchically organized does not really make sense. You can place a seed region in the frontal lobe at Brodmann area 8, and you will find Brodmann area 8 everywhere. But why is that? Because a lot of fibers converge onto that. So in order to understand the networks, you need to do something else, and that's from the same study. That's now coronal sections. That's in the MNI brain. That's axial sections. That's sagittal sections. And if you look here at number 4, that's again the VTA projection pathway, or SLMFB. And basically, in all the treatment regions that are basically shown here, you have the medial SDN. You have the anterior limb of the internal capsule. Here's where the inferior thalamic peduncle is. Here is the red nucleus of the stria terminalis. You see that the effect network is always addressed. That the reward network is always addressed, which we think is very important for our understanding. And in the due course of what we're looking at, we can actually find, even for the anterior limb of the internal capsule, and like a new corticopetal or bottom-up systematics, which basically say in the ventral part of the anterior limb. There's the effect and the reward pathway situated, medial effect and lateral more reward, although they are overlapping. You see the orange here is effect, and that's reward. And then you have the control networks. They're sitting up there. And we know if you do large lesions next to the caudate in the anterior limb of the internal capsule, then patients will be confused, at least for a while after that. And the reason for that is because that's where the control networks are, and you can really show them with stratigraphic imaging. And this has been proven also that you probably want to lesion in the anterior limb, in the ventral part more medially, and you want to stimulate more lateral to get a good effect, at least in OCD. That could check out to something which is more like a cartoonic. Again, you see that the reward and the effect network, they're playing against each other, and they're working with, and they're dysregulated to a certain extent. And if you carry that actually further, and then you can actually understand even how the always debated and always discussed corticostriatal or thalamocortical loop actually finds expression in there, only that this loop, the CSTC loop that is always said is important for OCD, for the hyperconnection of the thalamus to the frontal lobe and the striatum to the frontal lobe, that this is actually spread out between two systems, and it's not one loop, it's a loop that goes over two systems. So I think that's very, very interesting. We're doing medial forebrain bundle stimulation. I'm going to skip over this slide because it's just showing how we do that. We stimulate a region that is very, very close to the ventral tegmental area, just where the two parts ascend then to the frontal lobe. We are now using machine learning algorithms to guide our deterministic tracking. Deterministic tracking is unbelievably subjective, so we need to find something that basically helps you to do a more objective tracking of it, and that's what our tracking corridor basically subserves here. That's what we do in this work here. And actually, if you turn that around and look for your electrode that you've placed before, this algorithm can actually tell you that this electrode in a patient who did not quite so nicely respond to OCD DBS has basically an electrode that is sitting outside. DBS for the treatment of depression in the medial forebrain bundle is efficacious. It works rapidly. You can see how fast they go up within weeks, actually within days, if you want a closer look at that. And that is something that has been shown by other groups as well. That's the work from Alfenoy from the Houston group. We've recently presented our last results from the second trial that we did, which we performed as a gateway trial, 16 patients, and we had the same problem that the other trials had as well. Our placebo and the active group did not separate nicely. It actually happened in the last round with one patient, and then it would have been different, but it was not. But if you look at the long term, that's the response. If you look at the long term, what you actually can see is the whole group as itself is nicely responding up to 52 weeks. So SLMFB-DBS appears to work. There have been some benchmarking, research on that, which tells us that the medial forebrain bundle, amongst others, appears to be on the side where you think, yeah, it is a clear stimulation effect that should be working. I do not think you can draw any more important conclusions out of here, other that there have been some trials that were not too powerful and actually are crossing this line, which says that you're not quite so sure anymore about the effects. Summing up, it's very, very important to phenotype those patients. I've left out, for the sake of time, the phenotyping part of this section here. We need to look at the mechanisms. We are looking at animal models. We're now going into a large animal sheep model. We look at the human mechanisms. We're very, very interested in the networks and that in the bottom-up fashion. We need larger numbers to be treated. We have published around 30. Treated are around 50 after a recent telephone call with Alfenoy. The trial design appears to be key, and as long as we do not have at least more than 100 in one single trial, we will not have a solution to if this is working or it's not. It's a very expensive therapy. It's a therapy in development, and last but not least, there need to be consensus guidelines for psychiatry and neurosurgery. The psychiatrists do not yet see the real need, despite some of them, like I have the luck to work with Tom Schlepfer, who I present here. He's the psychiatrist in charge in Freiburg for the DBS patients. And that's my final slide. Thank you so much.